So, good afternoon again from Seattle. The third part of our topics for advancing spinal cord injury, making a real difference for our patients, is Dr. Brian Kwon. He's a professor of the Department of Orthopedic Surgery and Neurological Surgery at the University of British Columbia in Vancouver. He holds a number of other titles. I'm not going to read them in the interest of time. Dr. Kwon has gone where few others have gone. He's actually looked inside the spinal cord of real patients, and he can tell us new insights in terms of what is actually going on inside the spinal cord. Brian, please take it from here.
Jens, thank you, and everybody at the Seattle Science Foundation. We are really grateful that you would organize this webinar. A real pleasure to be here, and a real honor to be participating with the other speakers who are real leaders in the field. I'm going to talk about biomarkers of acute spinal cord injury, and really this is a talk that in many ways has been introduced by both Michael and Shekar. I think my topic is really going to be around the topic of neurochemical biomarkers for acute spinal cord injury, and we're going to touch upon, I think, a lot of the challenges that others have introduced in their talks, both Michael in his talk on the pharmacological trials and Shekar on his neuroimaging biomarkers for acute spinal cord injury. So I'm going to start actually just with the definition of what a biomarker actually is. And the biomarkers have been defined by the NIH as a characteristic that is objectively measured and evaluated as an indicator of normal biological processes, pathogenic processes, or pharmacological responses to a therapeutic intervention. And in biomedicine, there are some very common and very widely used biomarkers. And these would include things like the prostate-specific antigen, the troponin, or the CD4 count. And these are so commonly used in the disease entities of prostate cancer, myocardial ischemia, and HIV/AIDS that they almost become synonymous with these disease entities. Now, in the treatment of prostate cancer, myocardial ischemia, and HIV/AIDS, we will always do a history and clinical exam. And in combination with that, we use these biomarkers, the PSA, troponin, or CD4 count. And with that, we can predict outcome, guide treatment, and monitor the response to treatment. If you juxtapose that in contrast to what we have in spinal cord injury, in the absence of having objective biomarkers, we are really dependent on the history and clinical exam to predict outcome, guide treatment, and monitor our responses to treatment. Now, if we look at this clinical exam more carefully, and this was alluded to in some of the other talks, so we will examine individuals with acute spinal cord injuries when they come in to emerge on this, using this INSCI standard neurological classification scoring system. And this is a scoring system that measures motor function and sensory function throughout the body. And with that, we can assign an ASIA impairment scale, or AIS, of A, B, C, D, or E. Now, one of the major limitations for this is that, as Jim pointed out, the ASIA conversion is very difficult to predict these days, particularly if we do our baseline neurological examination early. And this was highlighted in the Staskus trial that Michael published a few years ago. Furthermore, if we were to look at ASIA motor score improvement, this is also highly variable within each of these ASIA grades. And this necessitates the large numbers of patients that we require for clinical trials. Michael referred to the 248 patients that were expected to be enrolled in the minocycline clinical trial and the 350 patients for the RISCA study. And this is because of the variability in the neurological outcome, even within these basic ASIA stratifications. Finally, this is a clinical exam, and so it does require us to be able to ask the patient to participate in these in the motor examination and the sensory examination. And this is obviously impossible to do in a large number of patients who come in that are multi-traumatized, that are sedated due to either intoxication or even just with the medications that we give them to control their pain. And we showed in a study that almost 30 to 40% of patients that arrive in emergency are unexaminable and for this reason unrecruitable into clinical trials. And Thomas, this speaks to one of the questions you asked Michael earlier about why is it so challenging to recruit these patients into trials? And it's because the number of individuals, or one of the reasons, is the number of individuals that can actually be recruited is a much different number than the total number that get admitted to the emergency ward. Okay, so this is a major problem for the testing of novel therapies for acute SCI. And this is where we see this as a major barrier for successful translation. Simply put, using this functional examination, many patients can't be examined, therefore they're not recruitable for clinical trials. Even when they are examinable, this classification is a poor predictor of outcome, and that's why we need so many patients to have adequate statistical power. And finally, we have only these functional readouts of outcome to tell us if the treatments are actually having the desired effect. So for this reason, we are struggling with this, what Jim described as this heterogeneity of acute spinal cord injury, for which we really assign an A, B, C, or D, but recognize that there is in fact quite a large spectrum of heterogeneity that is probably better described or could be more described in a more complex manner. So why biomarkers for acute spinal cord injury? To better characterize this heterogeneity, to more objectively stratify injury severity, to more precisely predict neurological outcome, and finally to monitor the biological responses to interventions that we may be interested in testing. So we have sought a biological assessment of acute spinal cord injuries by looking at not the spinal cord itself, but the surrounding cerebrospinal fluid. And by taking these CSF samples through a lumbar puncture and the installation of a lumbar intrathecal catheter, we have obtained serial CSF and blood samples from individuals with acute spinal cord injuries. So we have enrolled at our center and now across Canada and centers in the United States, individuals with both cervical and thoracic traumatic spinal cord injuries of ASIA A, B, or C severities. The lumbar catheter is installed and CSF samples are taken for a biochemical analysis. And we started this by evaluating these inflammatory cytokines, so things that mediate inflammation, and also some structural proteins that include tau, S100 beta, and GFAP. The major exclusion criteria in our study is the inability to conduct this examination. And so this actually rules out a lot of patients when you're not absolutely confident in the ability to do that baseline exam. So we initially started this and published our preliminary early results a number of years ago on CSF inflammatory cytokines and biomarkers of injury severity in acute human spinal cord injury, and continue to recruit patients for this type of investigation. And I'm going to just share some of our more recent analyses that we're just in the process of publishing now. So this is the status of the first 50 patients that we've recruited into our study: 32 cervical, 18 thoracic, a combination of ASIA A, B, and C patients. And importantly, we were able to follow these individuals for 6 months to determine whether or not they experienced ASIA conversion or an improvement in their ASIA scale, and a measurement of how much their motor score changed over that 6-month period. We were very diligent in our follow-up and achieved 96% follow-up in this patient cohort. And what did we find? So this is a somewhat busy slide, and it highlights it illustrates the concentrations of these proteins within the cerebrospinal fluid between the ASIA A, B, and C patients shown in red, blue, and gray. Now, these differences may not look very significant, but the scale here is a log 10 scale. So a difference of 1 point on this log scale actually represents a 10 times difference in protein concentration. So what we see here is that the concentrations of IL-6, tau, S100 beta, and GFAP were significantly different between the ASIA A, B, and C patients. when we were trying to then use these proteins to say, how do these proteins predict whether or not a patient would achieve ASIA conversion over that 6-month period? And in this case, ASIA conversion represents an improvement in their ASIA grade. Because none of the patients we saw an ASIA deterioration. So can these proteins predict ASIA improvement? And we see here that in red, those are the individuals that did not achieve ASIA grade improvement, and in blue, those were the individuals that did achieve improvement. And you can see across the board that the CSF levels of the proteins are significantly higher in those individuals that do not achieve ASIA grade improvement over that 6-month period. One of the things that, and again, if we get back to this heterogeneity concept and ask the question, well, can you use these then to try to distinguish between these individuals who are all ASIA A complete quadriplegics when they first come in? Again, we're looking to try to distinguish, try to discern some of the heterogeneity, try to find some of the biological factors that might predict why some of these patients with complete injuries might improve and why some of them might not. And so if we look at the 6-month post-injury status of these individuals and looked at those who did not convert versus those that did improve, all starting with an ASIA A, we found that IL-6 and S100 beta were significantly different between those individuals that started as ASIA A and remained ASIA A, and those that started ASIA A and became either an ASIA B or C over the 6-month period. Finally, if we look at the prediction of ASIA motor score improvement, so this represents how much muscle function individuals get back over time, we see that the CSF levels of all of these markers were significantly correlated with the motor recovery at 6 months. And we can use complex statistical modeling to try to model those that will and will not recover function. So this is a statistical technique in which we can actually identify those people in this valley here. These are the individuals that, based on their S100 beta and IL-6 levels, really did not achieve any significant motor recovery over the 6-month period. And where we would find this type of information useful is if we could have an individual that had these markers and demonstrated very poor predilection to achieving motor function, and then they did in fact achieve motor function with the administration of a novel neuroprotective agent, for example, it would help us interpret that that result was actually attributable to the neuroprotective agent and not just spontaneous natural recovery. So our results to date suggest that these neurochemical markers indeed hold promise for injury stratification and prediction of outcome after acute spinal cord injury. This we hope we can develop into a tool that will help in the early stages of clinical trials for stratification, prediction of outcome, and potentially also as a surrogate biological outcome measure to test the effect, the biological effect of these therapies. When we speak about future or ongoing work, this was a study that we began in Vancouver and have expanded it into multiple sites across the country. And these are some of our collaborators in Montreal, London, Ontario, Halifax. We've recently opened at UCSF in San Francisco and in Stanford. This is work that's been supported by the Mercanson Institute. We are in the process of also analyzing the blood samples from these individuals, because at the same time that we take a CSF sample, we also take a blood sample. And so we are currently conducting a parallel comparison of the changes that are reflected in blood. And finally, it is not certainly lost upon us that looking at these 6 proteins represents a very small subset of the biological responses to acute spinal cord injury. And we are in the midst of conducting a more expansive analysis of these very valuable CSF and blood samples, doing proteomics, metabolomics, lipidomics, and genomics. This is work that is being supported by the Brain Canada Foundation. And with this, we are hoping to analyze our samples. We're also analyzing samples from a large animal model of spinal cord injury. And our hope is eventually to put all of this information onto a searchable sort of library that can be used for further hypothesis testing the field, really as a resource to try to give the field more insights into the biology of acute spinal cord injury. So this is ongoing work. So it is an ongoing multi-center collaborative initiative. Our results to date suggest that these neurochemical biomarkers do hold promise for injury severity stratification and prediction of outcome after acute spinal cord injury. And again, we hope that this can be developed into a tool for early-stage clinical trials. So thanks very much, and happy to entertain any questions related to that.
Brian, thank you so much for sharing this exciting and very compelling data with us and these early insights. Dr. Fehlings, as lead investigator of the largest and exciting new international trial on riluzole and the management of acute spinal cord injuries, how do these insights apply to your trial, and how could this affect care in the near future?
Well, the use of biomarkers is an exciting strategy because it allows insight into pathophysiologic mechanisms. And it has the potential also to fast track early phase clinical trials. So for example, if one is investigating a putative neuroprotective drug, one might be able to fast track such a clinical trial by looking at biomarker readouts, which could be much more readily attainable. In addition, in the context of, say, a large phase 3 randomized trial such as riluzole, biomarkers could allow one to discern subsets of patients who might show differential responses to the drug. So typically, with any kind of a drug a therapy, and conceivably for acute spinal cord injury, there will be responders and there will likely be patients who are less responsive to the treatment. And so an examination of the biomarker profiles could potentially discern those subsets of patients that might potentially benefit from this type of a treatment. So I envision that the use of biomarkers would allow pathophysiologic insights, would allow us to discern subsets of patients that might be optimally treated with certain types of pharmaceutical approaches. And overall, it represents what I think is an important advance in the field.
Thank you, Michael. I have a question for Jim. So you live and work in one of the most litigious cities in the world, Philadelphia. How safe is it in an acute spinal cord injury to drain CSF and get a CSF sample out of a patient? Should we perhaps even think about opening up the dural sac to allow the spinal cord to expand?
You know, it's interesting. It's an interesting question because we're cycling around again. And actually, if you go back to the literature, there's a couple papers and some recent animal models that have shown that durotomy, or opening up the thecal sac, and even going one step further with even a myelotomy, might be more beneficial than we thought it was before. So I think we have to keep our, and I say this in a very litigious city, our primary goal is our patients, and we need to keep our aim at helping our patients. And I think Brian's, he's onto something here. I think it's a great idea. I applaud him for his work, and I think, full speed forward, he's doing this not as an experiment, but as an investigation. And I think it's a leap forward for spinal cord injury research. And actually, since you gave me the podium, which is kind of a mistake, Brian, and actually this goes back to you, Shekar, too. One of the issues with spinal cord injuries is it's not a discrete injury, and it kind of is a moving timeline along there. Brian, you picked out troponins, and all of us know that troponins usually peak and then they cycle down. So one of the things about troponins is we get 3 sets of troponins, and you showed 6 markers, which I thought were great. Are you there? Do you think you're hitting things? Because I imagine you're sort of randomly grabbing time samples, and how much more work do we have to know? Is each one a discrete curve?
Yeah, that's a great question, Jim. And when we first published our work, we actually showed the dynamic sort of range and how things were changing over time. And so we've actually focused a lot of our current efforts on the 24-hour time point, because we felt that that's a time point that it would be realistic to get a sample. We are actually taking samples, periodically over the first 5 days, and our nurses are, no matter when the drain goes in, they always go back to take the 24-hour sample. And so that there is some consistency with the time in which it happens, because you're absolutely right. This is a dynamic process, and there needs to be some standardization of the time. And in fact, in our experience, this is where we have witnessed some of the limitations of MRI, because the MRI happens when you can get an MRI, and it's hard to say, well, we're going to get an MRI at exactly 24 hours post-injury. So I think the timing of it is very important, because it is capturing a snapshot of what the spinal cord is doing at a specific point in time. We've also looked at area under the curve if you track things over time, and Steve Kasha has done some work similar to that. We think that if we're trying to develop it as a tool for trials, we need to develop something that's kind of a single time point marker, potentially with multiple components to it. Maybe some proteins. There may be some other small biomolecules that may be part of a signature that would help to discern some of the heterogeneity of the
Shekar, you've given a great presentation in the previous section on changes in neuroimaging for spinal cord injuries. A lot of the markers that Brian showed are anti-inflammatory. Where do you see the greatest intervention option? Anti-inflammatory management, like the previous steroid era, or membrane stabilization, like Dr. Fehlings as lead investigator, is showing with his Riluzole trial. So where's the main focus of pharmaceutical intervention? Anti-inflammatory or membrane stabilization?
Thank you for that question, and my compliments to Brian. That was an excellent presentation. I guess the answer to that question is, I think one of the things that has come out in this particular webinar, both with the approaches that Michael earlier alluded to in terms of the various types of clinical trials and their targets, the imaging data that seem to suggest that you may have a finite primary injury, but the secondary injury is certainly an evolving, it's got a dynamic profile in the injured patient at various times after injury, which is again brought out by the biomarker data that Brian just presented. So the answer to that question may not necessarily be one intervention, but different interventions. Eventually, I think maybe different interventions timed appropriately at the appropriate times might be the more appropriate way to look at it. So you may need an anti-inflammatory followed by a membrane stabilizer in some sort of a temporally worked out fashion that makes the most meaningful difference in what is likely a personalized injury. It's not necessarily a blanket C5 or a C6 injury. It's a personalized problem, and it may need a personalized solution.
Thank you. Let me ask Dr. Schildhauer to address Dr. Kwon. Dr. Schildhauer has one of the very revolutionary robotic spinal cord injury rehabilitation units, and I think he has a question for Brian.
Well, we all face the problem that we often don't know what the conversion rate will be, what patients will convert. The question would be, with having the morphological results from Shekar, your results, but also what we see in our neurorobotics is that the Asia classification may not be a sound basis for predicting the outcome. Is it time, in your opinion, to rethink that classification system? Do we have to rethink how to judge our patients early on, but also in the later chronic stages?
Well, I think that I mean, the Asia examination has great advantages in that it's widely used, understood, and you can be deployed in multiple centers with reasonable inter-observer reliability. And so, you know, it is what it is. I think that what we understand, I think what everybody appreciates, is that the heterogeneity of the injury itself far exceeds a 4-point ordinal scale. And you know, where I think that we will eventually move to is probably some type of a composite score where, you know, you may have some degree of a functional evaluation combined with, you know, potentially a neurochemical marker combined with potentially an MRI imaging sort of analysis of the cord, and with that be able to stratify patients, you know, in a more predictive manner. You know, one of the questions that we've, you know, grappled with is, you know, if you are using the biomarkers to compare baseline Asia severity or Asia grade severity, can you ever get any better than Asia grade severity? Really, I think where the value is going to be is if we can have a better way to predict outcome. That's where the big advantage will be. And so maybe, you know, their you know, Asia 1, 2, 3, 4, 5, 6, you know, scale versus Asia ABCD based on that combination of factors. But I don't know if you can really, at this stage, given the widespread use of the INSCI exam, really eliminate it. I think it's really part of the lexicon for clinical evaluation and trials for the near future, for sure.
Our chief of spine, Dr. Oskouian, has a question.
Brian, I have a question for you. I'd be interested to see the results when you guys look. I think it's ingenious to look at the plasma and see what, you know, especially in terms of proteins, rather than this. I think the spinal fluid is one aspect of it, but are there certain proteins that you guys are looking at?
Yeah, so we're doing both, to be totally honest. We did blood sampling and blood analysis early in our CSF analysis, and we found that the levels were in many cases not just an order of magnitude, but in some cases many orders of magnitude lower in blood than in CSF. This is not to say that they may be uninformative, so we are still evaluating that. And there may be other biomolecules that could be informative, such as microRNAs or other small molecules that we might pick up in our metabolomic screening. So, you know, in truth, when we are trying to be rather agnostic about what the best biomolecules are, but we're definitely interested in doing that parallel analysis of blood and comparing it to what we see in the CSF.
Great talk, Brian. Brave new and audacious new insights. Congratulations on your work and that of your group. I want to thank our colleagues from Philadelphia, Wisconsin, and Toronto as well. This concludes the third part of our section. Thank you very much.